Hello, my
Hi,
name is Eric Soy. I am based out of Cold Harbor Lab, and I am the executive editor of Life Science Alliance. I'm joined here by Charna Dibner from the University of Geneva. Welcome.
thank you. Hi.
Hi. So the main focus of your research is to identify the molecular basis of circadian rhythmicity in both human peripheral tissues as well as in mice. And I know you've examined this rhythmicity under both physiological conditions and then also with a particular emphasis in models of obesity and type 2 diabetes. I was interested first if we could start off by asking you what sparked your interest in this area of research?
Right. So, yeah, thanks, Eric. So until now, indeed, we are working on the combination, and I think it's very important to combine genetic mouse models and to do to translate it to human peripheral tissue to the extent that it can model the disease in order to have the combination of these two approaches. I think neither is perfect, right? So I do not really think that we believe that OB, all these mice, models, they really perfect. They're not a perfect model of human diabetes. Neither it is when you look into human peripheral tissues. But first, we cannot do better, right? We cannot do it in vivo in humans, this kind of circadian research. For sure, not on endocrine pancreas. And I think the combination of the two works perfectly well. So the thing that we actually were particularly interested when we started to look into the clocks in endocrine pancreas is this heterogeneity of the cells within the pancreatic islets, right? Because it's quite unique as a model. You have a three-dimensional ball, basically, where you have different endocrine cells. So we focused on beta cells that secrete insulin and alpha cells secrete glucagon. So the first highlight was that, first that we could measure this clock. So now we have a combination of genetic mouse model where we can separate nearly pure population on alpha cells, and the technical difficulty here is just they're very scarce. But with the reporter that we have, it's just, it works really well. And then we could categorize the clocks, and what came as a surprise is that they were not exactly aligned. So that was the first outcome of this research. And then why this is important, actually, beyond the chronobiology field, is because what we also found that this cell autonomous clocks in alpha and beta cells, they are super important for temporal coordination of secretion of insulin and glucagon. So that was the outcome of our first work. And now what we try to find, because basically what we've shown is that this is the first step, these are clocks, right? And then there are hormone secretion, which are just downstream the pyramid. So now we are trying to, so the physiological phenomenon is clear, and this is very interesting, that you have basically the secretion, temporal secretion of insulin and glucagon that is driven by these molecular clocks in alpha and beta cells, and they're not exactly aligned. And the meaning of this probably that it helps the body to anticipate the activity phase, to be able to jumpstart when the glucose, the food is coming, right? So that's what we think. So now what's left completely as a black box is the whole thing in between. So we are trying to dig into molecular mechanisms. For example, we are studying the role. So we looked into transcriptome so far, but of course, transcription is something that is the easiest level to look, but this is not yet everything. So now we are doing also protein levels. So we're doing proteomics, probably phosphoproteomics in alpha and beta cells. And we're also looking into microRNA. So we would like to connect between the two dots to look into mechanism. So that is one, I think, exciting direction. So now, then the second message was, what is directly related to diabetes. The main question is how to make beta cells to regenerate. So what we have here is the mouse model of basically, which is close to type 1 diabetes, where we can destroy at once about 80% of beta cells. And again, so the work that we published just end of last year was the two main messages were that first, this compensatory regeneration of beta cells that is induced by this destruction is rhythmic. So that was really cool. And it also showed the field, basically, also when we work with people who are non-chronobiologists, it's important to show that it's really, because mice are nocturnal and the researchers are diurnal. So, if you don't inverse them, sometimes you really don't fall in the optimal actually window of the activity of certain phenomenon. And that's what also I think it's important message that I try to communicate usually to the islet non-chronobiologists. So the compensatory regeneration in mice is really highest in the middle of night when the mice are most active. And second important message was that we need the functional clock for this phenomenon to happen. And so now, because you asked me before these questions about how we can do this recording maybe in freely moving mice. Mm-hmm. So we are using this model of beta cell compensatory regeneration indeed to look in the record in vivo in freely moving mice. For example, we can do the liver recording. And we can look into the liver clock and different other organ clock in the context of this, what we call type 1 diabetes-like model. We also try to translate now this model into clinics, and we are looking in parallel into continuous recording of the glucose levels in type 1 diabetes patients. And so we hope to combine these two research lines.
Yeah, that's really interesting. So I was curious about when you do the beta cell depletion, is there any effect on the mouse feeding behavior or feeding pattern after those experiments, or either with or without BMAL1, or you don't see an effect?
I mean, we did not look for this effect yet because we put them on night feeding. Let me see. So that is something that we do not address directly. My hunch is that they will probably, well, when we did it, clearly when we did this experiment on BMAL knockout background, they have a clear feeding phenotype. I mean, they do not have a feeding phenotype, and we try to, to compensate for that by introducing night feeding, because we would like to keep, we would like to ask one question at a time. So it is an interesting question to ask. We should maybe design that, at some point,
And then in your talk, you talked about some of the work you've done with human pancreatic islet cells from type 2 diabetics, and you show that the circadian oscillators exhibit a disrupted pattern. I wonder if you could talk a little bit about that work and kind of, what's next with that.
Yeah, thanks. So indeed, we saw, and that also came almost as a surprise, because the human tissues, they have such a huge heterogeneity that to find the effect that will override the heterogeneity between the subjects, I mean, the effect should be really massive, that you can capture it between the different groups. And that was really also, the most important outcome, first important message from that work, that was recently published. And so we could zoom into it sort of, because we did time-lapse recording in individual alpha and beta cells within the context of human islet. And we could really see that both circadian amplitude and synchronization between among the cells in alpha and in beta cells was disrupted. And then, so another very important message was that beyond the disruption of insulin secretion in type 2 diabetes, that is definitely hallmark of type 2 diabetes, that was not new, but nobody ever looked before that at the continuous secretion. So what we found is that also not only the absolute levels of insulin secretion, but also its temporal pattern was disrupted. And based on our earlier work, because what we could, I mean, of course we cannot do transgenic humans, but what we can do in this culture is that we could, using siRNA approach, we could knock down to large extent the cortical component clock, and then the clock sort of flattens. So that was our earlier work, from which we learned that this also impacts in kind of similar way on insulin secretion. So you do need functional clock, and it could be, so from our last work, what we sort of suggest that probably part of the pathogenesis of type 2 diabetes is that the clock gets flattened, that it's less functional. And then we could boost it, and along with that, we could also, to some extent, restore proper insulin secretion with the clock modulator nobility that boosted the amplitude. So that I found very interesting, because then by boosting the clock, you actually also again, this is, we will not say that this is now the new treatment, but this is something that opens at least the perspective to explore it, maybe in combination with classical anti-diabetics, you know, and classic GLP-1 antagonists and so on. Maybe one can also try the clock modulators potentially.
Wow.
So
Yeah,
to continue on that, it's so we first want, because we now found that this is very clearly that the insulin secretion is connected to functional clocks in human islets. We also would like to know why is that. So we look potentially. What I think one thing that is really interesting is lipid homeostasis in this context. And early on, we published works that clearly shown that in human skeletal muscle, and that we show in vitro and also in biopsies, sort of ex vivo, in serial biopsies. So we have shown that that really about 20% of, we use shotgun lipidomics, where you can measure over 1,000 lipid species, membrane lipids and signaling lipids, and triglycerides also. So we've shown recently that in skeletal muscle, you have about 20% lipid species that are oscillating, and they also depend on functional clocks in human muscle. So now we are, along with these lines, we are currently exploring the lipidome in human pancreatic islets in diabetic context and in islets from healthy donors. And we would like to see how does that. So we do see already, I can tell you that we do see the arrhythmic lipids in pancreatic islets, and there is a massive change in the islet in the context of diabetes. This mostly affect sphingolipids and deoxysphingolipids. And this is very interesting. And we are also doing functional experiments, how by modulating sphingolipid synthesis pathway, we can influence insulin secretion. So this is something I think very exciting as direction. Just to, I mean, for sure, this is interesting to see how the clock affects lipids in human pancreatic islets, but how also there is also one perspective again with type 2 diabetes. So this is something. And then again, we're looking mechanistically, because still we do not clearly connect the dots between how the clock really affect. So one element we do have, we did TIRF microscopy, and we know that the effect is going at the level of exocytosis of insulin granules. One other thing that we have preliminary that maybe they do not mature as well. But so I think to explore this mechanism between the clock, what's between the clock and insulin and glucagon secretion, this is an exciting question, and we're trying to develop.
Yeah, that's really interesting. And then just to try to extrapolate this even further. So I believe in shift work, right, and people has been linked to insulin resistance. Do you have any guess if some of this mechanism you revealed is related to that?
Right. So people who followed now Cold-Spring-Harbor laboratory from Geneva, for example, we call this situation circadian misalignment, right? So yes, I mean, that was clearly shown at the level of insulin resistance. That was done in a very well-controlled, we call it laboratory studies of circadian misalignment. Frank Shear, for example, published very elegant works that it is enough to either sleep deprive or misalign people. I mean, they did simulated shifted schedule, and that really there is transient insulin resistance that developed after surprisingly very short period of circadian misalignment. So we should all be aware of that. Now, at the level of insulin and glucagon secretion, this is pretty difficult. I mean, of course, we cannot see what's secreted. I mean, to my knowledge, that was not really very clear, because I do not think that somebody did the really circadian profile, or at least I'm not aware. Maybe somebody did it, I'm not aware. For insulin secretion, I would say this is pretty tricky to dissect also, because clearly we cannot have isolated. So what's cell autonomous, it will be very difficult to measure. Now with glucagon in the blood, it's also technically very difficult to measure. We are now trying, I mean, of course, everyone sort of measuring that, but there are many technical problems with that. It really depends how well the samples were stored. Now for insulin, I'm not aware of something very clear in this direction. So I do not know if short misalignment can directly have an effect on insulin. For sure in insulin resistance, that is clear cut and scientifically proven. Now it is also clear, I mean, that there is a correlation between long-term shift work and obesity and type 2 diabetes. So that is very clear that people who are subjected to, and now we're trying to look a little bit differentially on the cohort of shift workers and to see what makes, because some people tolerate it less well and some people tolerate it better. So I think the $1 million question is just to identify what makes you to be resistant, what makes your metabolism to be resistant to this chronic shift work. So, but what we know so far, I'm not aware of any, let's say, molecular study. Doesn't mean that there are none, but I'm not aware that would connect the shift work to insulin secretion. And that is also not quite tricky, I would say.
Yeah.
But for insulin resistance, this is clear cut. And for sure, I mean, if you stay misaligned for long, this insulin resistance may become also chronic. So that is very clear.
Very cool. Well, I think that's the time we have. So I really want to thank you for sharing this interesting work with us. And I thank you for your time.
Thank you very much, Eric.
Okay.
Bye.
Bye.